Hello and welcome to my next video on variation. Now the first page is just AS stuff, so you should all know this. Variation is the differences that exist between individuals. And you have continuous variation, that has a range of things, so height, weight has a complete range with infinite number of intermediates. You can have two or more genes will provide the effect, and each gene provides an additive component. Now what that means is that each gene you have gives an extra kind of boost to that characteristic. So height, if you have 3 genes, it might mean that you're taller or something. Now in continuous you can have polygenes. Now a large number of different genes may have a combined effect on their phenotype. These are known as polygenes, and the characteristic they control is described as polygenic. The genes are unlinked, they are on different chromosomes, but all together combined to do an effect. And then this is the kind of newer thing, basically saying different alleles at same gene locus have a small effect. That means if you have 2 alleles so they're at the same point on the chromosome, but they're different alleles, they will only have a small effect. There won't be a huge difference. That'll make more sense when I talk about discontinuous, because it's kind of the opposite. Discontinuous is distinct categories, you can't have intermediates. Now this is different alleles at the same gene locus have a large effect. Basically, the best way to explain it is using this one, because you can say, think of eye colour. You can have a brown eye gene and a blue eye gene, and each of those genes can have a large effect. So if you have a brown and a blue, then you're going to have brown eyes. If you have 2 blues, so it's different alleles but the same gene locus because they are the same gene, then you will have blue eyes. So basically discontinuous refers to dominant, recessive and co-dominant genes. Continuous doesn't. So if you have 2 different alleles, it doesn't make any difference really, or not a huge one. And different gene loci have different effects. So if you have more than one gene that is coded, that codes for something, and at different positions on a chromosome, then they have different effects on that characteristic. So for example, with eye colour, it's actually you have a few layers of eye colour which make up your overall eye colour. That's why not everyone is just brown or blue. So you could have one gene that says you're going to have brown eyes, but somewhere else on that chromosome you could have another gene which says blue, and it combines together to form a kind of green colour. Something like that. And the other important thing is that your genes might say that you're going to be incredibly tall, but if you don't eat and become malnourished, you're never going to grow to your full potential. So, while discontinuous is generally genes, continuous is environment, there is a crossover as well. So you could have the genes to be tall, but without the correct environmental conditions, you're not going to present those genes forward. Now I enjoy this. The Hardy-Weinberg principle is actually some real mathematics in biology. Now it looks confusing, but it's very simple if you just remember stuff, as all biology is really. You have 2 equations which you will be given. P plus Q equals 1, P squared plus 2PQ plus Q squared equals 1. And if any mathematicians rather, you can see if you square P plus Q, so P plus Q in brackets squared equals 1, you get the second equation, for anyone who cares. Now, P is the dominant allele you have. So if you have a brown-eyed gene and a brown-eyed allele and a blue-eyed allele, brown is P, Q is recessive, so Q is blue. And then P squared is the actual dominant characteristic. So if you are double dominant, so both you have a brown eye and a brown eye gene, you are P squared. If you are Q squared, you have recessive, so blue eye and blue eye. 2PQ is carriers. So that means if you have 1 brown, 1 blue, or 1 blue and 1 brown. And the way you do this is, now it's explained awfully in the book, and I can't actually see anywhere else that's written the rule, but I personally, I'm claiming this rule as my own. The anti-clockwise rule. You will be given a number, so 1 in 5,000 people have this disease, and it will be a recessive disease. So that means it is only carried on the recessive gene, allele rather, which means that it must be double recessive to be shown. So 1 in 5,000 people have cystic fibrosis and it's a recessive disease. So in a population. That means you've got Q squared. Q squared equals 1 in 5,000, which equals 2 times 10 to the minus 4. Then, what you do is you work out, you want to go in a anti-clockwise motion. So you've got Q squared, you then want to work out Q. Because if you're looking that kind of anti-clockwise movement, that's what you get next, Q. So what you do, square root 2 times 10 to the minus 4, and you get that number. Then, next you want to do P. P is 1 minus Q, if you just do a bit of rearranging. So P equals 1 minus Q, and equals that number, 0.9859. Now you've got P. Now there are two methods. Just for a bit of helpful remembering, if you follow the anti-clockwise rule, the next thing is P squared. So we're going to do the arrow going along, not diagonal. P squared equals that, you just square the number. You then want to work out 2PQ. That's next in the anti-clockwise. So you do 2PQ equals 1 minus Q squared minus P squared. Or I've done P squared minus Q squared. So you can see you've got P squared, 1 minus that, minus Q squared, which we had at the beginning, equals that value. Now if you do it the other way, if you go back to working out P, we've worked out Q, we've worked out P. So to get 2PQ, you just times P and Q together and times that by 2. As you see, they give the same result. Now I would suggest it is slightly better to use the work out P, then P squared, then PQ method, because you are just ever so more likely to get a very accurate result. With the 2PQ straight away method, you can get a very small error. Now, it's important, when you're doing this, write down rounded values, but when you calculate it, do not round your values where possible. That's why the 2PQ value can just come a little bit off if you go straight from P to 2PQ, because you can't store both those exact numbers in your calculator, you can only store one. With the P squared to 2PQ method, you can store all of them, and I'll show how it works. Now, I can't exactly show you my calculator, but Q squared, you type in 1 divided by 5,000, get that value. You then just do square root of the answer. So you just literally type in square root answer, you get that value. You then do 1 minus answer, will give you the exact value of P. You then square that answer exactly, giving the exact value of P squared. And then, since Q squared was already an exact value, if you do 1 minus P squared as an exact value, so 1 minus answer, minus 2 times 10 to the minus 4, you get the exact value of 2PQ. Then, I hope that makes sense. It's quite, yet again, maths is always hard to explain, but I hope you can see what's going on. Please feel free to email me on that. Just practice. I suggest get the CGP textbook, as it has a really good section on the Hardy-Weinberg principle. And then once you've got that value at the end, what you do is 5,000 times, so your original number population size, times your final value equals number of carriers. In this case, 139. Now, the other thing they might want you to do is work out percentage. Now, to do that, you just want, if you want a percentage, just times your final value by 100. Now, one thing to make very clear, this is where the rounding actually comes. You don't want to round. My final value I've given there, 0.0279, is rounded. Now, if you don't round it, and times it by 5,000, you get 139.4, which rounds to 139. If you times it by the rounded value, 0.0279, you get 139.5, which rounds to 140. Therefore, you are wrong. You're off by one person. So it's just important, don't round. Keep it stored in your calculator and go through. That's why. So here, if you used the 2, went from P to 2PQ straight away, you would have got the answer wrong. If you use the exact value method, you're going to get it right. So don't round if possible. Now, the problem with the Hardy-Weinberg principle is that it makes 4 assumptions, which is bad. You don't want to make assumptions, but you have to, which makes this kind of whole thing a bit pointless. 2 of the assumptions are fair enough, you need them. The other 2 are just make this whole thing pointless. The first 2, the population is very large, So you have a completely huge population, so you remove sampling errors. Fair enough. And the mating within a population is random. Yet again, fair enough. If you're controlling who mates with who, then of course you're going to get, very exact results. So those two are fair enough. But these two, there's no selected advantage for any genotype. So for example, I'll use giraffes later for something else, but if giraffes with shorter necks, let's say that's caused by a gene, giraffes with shorter necks are going to die. Because longer necks mean you can get the food. So that's a selective advantage, which means Hardy-Weinberg doesn't work for them. And the other one, there is no mutation, migration, or genetic drift. So it basically means the kind of genetic population is exactly the same and never changes in terms of mutation or genetic drift, which is rubbish because that will never happen. You will always get mutation, migration, genetic drift, which means this whole principle is pointless because it won't work. I'm sure there are some examples where it can work, but generally you're not going to be able to control something enough to remove the chances of mutation, genetic drift and migration. So that's the problem with the Hardy-Weinberg principle, but if you're asked to do it, that's how you have to do it. As I say, get the CGP textbooks. The textbook is awful for it, the R textbook. There's also quite a good example in the revision guide as well. So, now we're going to look at the roles of genes and environment in evolution. Now, two key terms, environmental resistance and selection pressures. Now, we're going to look at this more in the ecosystem sustainability module later on. But environmental resistance basically is, as a population grows and grows, eventually it will reach its carrying capacity. That's the maximum size it can be and still survive, because of, nutrients and space available. Now, the environmental factors will limit the population growth, and these factors offer environmental resistance. It's the environment kind of stopping the spread of the population. So if there is, let's just say, randomly, there is 100 foods in an area. One food per animal for life. I know that doesn't make sense, but just kind of as an idea. So one food will feed an animal for life, and there are 100 foods. That means the population can reach 100. Any more than 100, then they will not be able to fully feed themselves. So food becomes is kind of providing environmental resistance. Right. And we'll look at more about population fluctuation in a later video. Selection pressures. Now, selection pressures is basically something which will select which organisms are likely to survive. So, with giraffes, I'm going to use giraffes because I like giraffes, a selection pressure would be, well, they have their neck size. The longer necks will be able to reach the trees. So the trees are becoming the selective pressure because they are encouraging a longer necked giraffe to survive. So it's kind of selecting the longer necked giraffes because of the height of the trees. That's what selection pressure is. And these selection pressures lead to two types of selection. Stabilizing selection, directional selection. Now, stabilizing selection means that basically the mean kind of trait in a population is the good one. So if you have wing length in birds, really short wings, they won't be able to fly or have as much ability to control themselves in the air. So that's bad. Too big wings and they'll become heavy and won't be able to do the same sort of acrobatics, and they will also die. So the ones with the mean wing length, the best mean wing length, they will survive. So the ones with extremes will die. So you get the range of the population decreasing and becoming much more closer to the mean. As you see here, the original is the one without the kind of dashes on it. So it starts off with a reasonable width, and then after stabilizing selection, much smaller width. The other one is directional selection. Now this is where giraffes are good again. Now this means that an extreme is favoured, and that will continue going. So giraffes, the ones with short necks won't do very well. The ones with long necks will do the best. The one with medium length, so the most common neck length, will do all right, but not amazingly well again. So it's going to favour the longer neck. So over time, there will be a shift to one of the extremes. Equally, it could be weight, and you want a smaller weight is better, so it'll go the other way. I've done it a bit extreme here. It wouldn't actually end up going so far to the right, but you can see, you get the idea. The dashed one has moved on because the selection pressures have deemed it that the more extreme animal will do better. Same like the thing about intelligence. Something that is more intelligent. I'm not talking about, just, like book smart. I mean, just generally, an animal which understands not to run up to a predator is going to do better. So it's going to favour the much more clever animal. That's just an example. Genetic drift, something I also think isn't greatly explained in the book. Basically, genetic drift is the change in allele frequency, and it's by chance. So here, I've shown a really weird diagram. You have 4 black alleles, 4 white alleles. Now this isn't, it's just one's coloured in, one isn't. And over time, just by chance, because by interbreeding, you will get a change in allele frequency. So in this case, you then get 5 and 4, then 7 and 1, and then just completely just 8, which means there's no chance that white one will come back, which means you've eliminated the white allele. Now, it's in the book, so I suggest pause the video and get the book so I can explain it using the book, because it works. The book doesn't explain it well, but it's got a good example. Now, I hope you have your book. If you look at the example on page 141, you breed with a heterozygous parent of capital A little a times capital A little a. And then let's say they have 2 children. That means the population size hasn't changed. Now, the possible genotypes of those children is big A, and big A, and you can see all the different ones there can be. Now, you can then work out the probability of each one occurring if you by drawing a Punnett square. The chance of all of these ones, the chance of big A occurring is a quarter, the chance of little a occurring is a quarter, and the chance of a big A and a little a is 2 quarters. Now, if you look in the book at the probabilities, this is the probability of each pairs coming. Now, to do probability of this and this occurring, so AA and AA, you times the 2 probabilities together. So the probability of AA and AA is quarter times a quarter equals a 16th. Now, big A little a and little a is 2 times a quarter equals 2/16, because there are 2 choices of having a big A and a little a. If you look at this, you've got all the different probabilities. Now, let's say, the probability is randomly by chance. That is the probability of each one occurring, but which one actually happened is by chance. So let's say by chance, big A and big A occurred. Now, instantly, you've lost the little a allele. So there has been genetic drift. You've only got 1 allele afterwards. That's what it basically is. And so you've lost the number of possible phenotypes as well, because it's gone from 2 phenotypes of a dominant and a recessive to just 1. And that so will always be the dominant. And I hope that makes sense. I think the book explains this awfully. But yeah. Basically, genetic drift happens in small populations because the probability of them occurring is very, is high. It's reasonably high that you will get in an equal sized population after, in the next generation, you will lose an allele. Now, imagine if it was 3 children. Well, then you've got, 0.25 times 0.25 times 0.25. So you've now got 1/64 to have all dominant. So it becomes much more unlikely. So the bigger the population, so for example, if we then get to 6, 7, all the way up to 10, you've then got the chance of 1 over a huge, we're talking millions of that thing. And that's just with 10 children. So it is only happens in small populations that genetic drift can really occur. Hope that makes sense. If not, again, please ask me. It's just quite a few stuff in this little chapter isn't done very well. But basically, it's that in a small population, you have a probability that you could lose an allele, and since reproduction is by chance, then you can lose that allele, and you then remove a phenotype. Equally, as you see in the example I've drawn on the slide, you have, by chance, you've got a change in allele frequency of just from 4 to 4 to 5 to 3. But if the same change keeps occurring, that's how you can lose one as well. So it could take a number of generations. Right, natural selection and artificial selection. Now we've done natural selection at AS. It's very simple. It's just random mutations cause adaptations. The adaptations that make organisms survive will mean that they are passed on, so that's evolution. Please watch an AS video if you don't understand that. It probably is in biodiversity in AS. Artificial selection is when humans select organisms with the characteristics they want. They allow the organisms with good characteristics to breed and the ones with bad characteristics to not breed. That's artificial selection. Now, I'm going to show you an example. You do need to know this, and it's about wheat, bread wheat. So, we start off with wild einkorn. Now, this is with the alleles AU, AU, and it has 2n equals 14. So there are 14 chromosomes, 7 pairs. Now, we domesticated them and use artificial selection. So this now alters the phenotype slightly because we're making sure we're getting the characteristics we want, so perhaps a dominant ones rather than a recessive, but the chromosome number is the same. So we've artificially selected them. Then we breed them with wild grass of the genotype BB. They also have 2n equals 14, so 14 chromosomes. We breed them together and they form an AU and a B. Just as you would expect if you do a Punnett square, you get an AU and a B. But this organism is sterile. It cannot reproduce. But then there's a mutation which causes the number of chromosomes to double. So you're left with AU, AU, BB, and you've got 28 chromosomes. Now, plants can do this. Animals cannot, but lots of plants, particularly grasses, can undergo polyploidy, which means they can increase the number of chromosomes in a cell. Now, with AU, AU, BB, which we call Emma wheat, we combine that with goat grass, which is DD, which has a normal number of chromosomes, 14. Mix them together and you get AUBD, which has 3n chromosomes, as you can see. But then again, this is sterile. A mutation causes it to double, and you get the common bread wheat, AUBD, and you've got 42 chromosomes, and it's polyploidy. But it has all the characteristics we want. I'm not 100% sure what the actual what the characteristics are we wanted from these wheats, but it's probably something to do with size, so kind of getting better sized, better tasting, things like that. Oh, here you go. It actually says things like resistant to fungal infections, high protein content, straw stiffness, that's the stem, resistance to lodging, that's stems bending over in wind and rain, and increased yields. That's what you want. Again, hope that makes sense. And now species. What is a species? We've done this at AS, but that's the one type of species concept called the biological species concept. That is a group of organisms that can interbreed to produce fertile offspring. The good old definition we know and love. But there's another one we can use called the phylogenetic species concept. Now this is a group of organisms that have similar morphology, shape, physiology, biochemistry, embryology, stages of development, and behaviour, and occupy the same ecological niche, and a niche is its role in the ecosystem. And the big thing about phylogenetic species concept is looking at evolutionary relationships, particularly on a genetic level. So we have a calculation. Divergence, so how different a species are, is calculated by the number of bases substituted from the original kind of code divided by the number of base pairs you are looking at. So in this case we have 9 bases, A, C, G, T, A, A, G, C, C, and then one is changed, and that's the T to an A. So one base has been substituted over 9 times by 100, so there's 11.1% divergence. Of course, it would happen on a much bigger scale, so you could get, 3 bases changing out of 2 million, then you have a very small percentage divergence. The ones that have the closest species percentage divergence are the ones which are more closely related. And we can use something called cladistics. Now, cladistics is the hierarchical classification of species based on their evolutionary ancestry. Now, it's different from taxonomical, which, if you remember, is kingdom, phylum, class, order, family, genus, species, because it focuses on evolution relationships rather than similarities between species. It plays great importance on quantitative analysis. It uses genetics, DNA and RNA sequencing, uses computer programming, and the important thing, it doesn't make a distinction between extinct and extant species, so living and ones that have died out. So you can still be related very closely to a dead species, more so than you are to a living species. So we are more related to Neanderthals than we are to blue whales. In taxonomy, in the biological species concept, it kind of focuses on living animals. And here is a kind of tree of life where you need to know how it works. Now, the plain circle with M next to it is a monophyletic group. That is where all the organisms in that group share one common ancestor. As you can see here, they share one right at the edge of the circle. I've drawn a big dot at the common ancestor. Now, the kind of circle with some lines through it with PO, that is a polyphyletic group. That is when you have 2 organisms who do not share a direct common ancestor. So as you can see here, these 2 animals are related, but do not share the common ancestors I've shown by either the square or a triangle. So that's a polyphyletic group. And finally, the dotted line with PA is a paraphyletic group. Now that means that it has a number of species which all have the same common ancestor, which is shown by the kind of oval, but don't contain all the organisms in that group. So here I've missed out the polyphyletic group. So while all the organisms in the paraphyletic group share the same common ancestor, not all the organisms with that common ancestor are included. So I hope that makes sense. Now, there are problems with both concepts. Now, the problems with the biological species concept is that the organisms might be extinct, so you can't tell if their reproductive behaviour. They might reproduce asexually. Now, if you have to interbreed to produce fertile offspring, asexual animals or organisms don't, rather. There might be practical and ethical issues involved. You can't see it. You can't see if some organisms reproduce successfully in the wild due to geography, and you can't study them in the lab because it's unethical. But there's also some problems with the phylogenetic species concept. The main one is there's no cut-off point. So we say, it's based on perhaps percentage divergence, but what is the value where 2 species become the same? Is it 0.00001? Is it 1% even? So how can you actually tell which 2 are a species? So for example, humans and chimps are 94% of our DNA exactly the same. Is that not close enough to be the same species? That's very similar. So finally, speciation. Now this is the process of creating a new species from old ones. And we've done this at AS as well. So I'm just going to very briefly go over it. There's geographical isolation, which is perhaps if you're on this kind of island, big island with 2 big hills. A flood happens, water separates them, so they cannot no longer cross. So you kind of geographically isolated, so only half the population can breed with each other. They'll create mutations and eventually become new species, so they can't interbreed with each other. Reproductive isolation, that is where 2 organisms are together but physically can't reproduce. So for example, let's say a bird is doing a mating dance and creates a new mating dance, and only half the population like it and only half the population will mate with it. Well, instantly it's reproductively isolated itself from half the population. But then when it has children, its children might copy the same dance. And over many generations, half the population will do a certain dance, half the other population won't, so they won't mate, and then they're different species. So thank you for watching. Sorry for the long video. So we have continuous and discontinuous variation. You have the Hardy-Weinberg principle. Practice that. Genetic drift, which is change in allele frequency. You have natural selection, which is how evolution works. Artificial selection, which is what humans do. What is a species? You have the biological species concept and the phylogenetic species concept, and you have speciation caused by geographical and reproductive isolation. So thank you for watching. I hope that all makes sense. Some reasonably confusing topics in this section. It's not, it's not really gone into a huge amount of detail, but they can be a bit confusing, especially because they're not done very well in the book. So please feel free to email me or leave a comment if you don't understand something. Always leave feedback. My rule on videos is I do not care if I get dislikes, because that's someone's opinion. Please can you say why you disliked it so I can improve, because I want to help as many people as possible. And thank you for watching, and goodbye.